Hello, it's a beautiful late fall Sunday afternoon on the Caltech campus. Because it's Sunday, not many students or faculty members are around. There is the entrance to the red door. We can't see its red door from here, which is very much the focal social point of the Caltech campus. It's a cafe. Here is the bookstore. And a little further around, we have the Olive Walk and the Student Center and the beautiful sycamore and olive trees. Some of these scenes may be familiar to you from the series Numbers, which was filmed on the Caltech campus between 2005 and 2010. Indeed, Cal-Sci, the fictional place where Numbers takes place, is actually a pseudonym for Caltech. So let's get started. And fittingly, since the red door is the nerve center of the Caltech campus, we will be discussing the center of the nervous system today, the brain. So this is an introduction to mammalian brains, neuronal circuits, neurons, and synapses. First, a little bit of an anatomical orientation. The central nervous system consists of two major parts, the brain at the top and the spinal cord projecting out from the brain. The front of the brain is the rostral direction, the back of the brain is the caudal direction, the top is dorsal, and the bottom is ventral. Now, an important concept is that specific brain regions are often the seats of specific functions. So, for instance, here at the top, we have motor cortex controlling movement just forward of the central sulcus, and just in back of the central sulcus is sensory cortex, part of sensory cortex, providing sensations. Back here in the occipital lobe at the caudal part of the brain is visual cortex, and here in the rostral part of the brain is so-called prefrontal cortex, and part of the function of prefrontal cortex is judgment and higher neuronal functions. The memory is laid down at first in the hippocampus, which looks very much like a seahorse, this yellow part. Motor coordination takes place in the basal ganglia and to some extent in the cerebellum. And an important part of drugs in the brain, the reward pathway, is shown here in a purplish. That's where nicotine and dopamine and several other reward transmitters play a role. Now, if we look at a simple circuit that involves the periphery, the spinal cord, and the brain, when you get your finger stuck in a door or a car door slams on it, the pain sensations travel toward through the nerve roots into the spinal cord, where they synapse onto neurons, which then project in an ascending fashion. So we call projections that go to the brain as ascending. Most sensory messages synapse in the thalamus, go to sensory cortex, and then we start making the decision to move away from the pain. So here we go to motor cortex, back down to thalamus, and then a descending pathway back to the spinal cord to a motor neuron, which tells the finger and the hand to move out of the way. So this would be a simple pathway in the central nervous system involving neurons and synapses and a loop. Spinal reflexes, for instance, the knee-jerk, are a bit simpler. They involve just two neurons. And so here is the physician's hammer stretching the tendon, and that signal comes from a special kind of muscle fiber. The spindle goes into the central nervous system, into the spinal cord through the dorsal root ganglion, synapses onto motor neurons. Those motor neurons come right out and contract the muscle and make the leg jerk up. Actually, there's a very interesting additional pathway which inhibits the opposite muscles. So if the leg is extending when the knee jerk takes place, there's also inhibition of the flexor muscles. And this reflex acts almost completely on its own with no descending control from the brain. Interestingly enough, here the sensory neurons act like strain gauges wrapped around these special muscle fibers. So we have a sense organ again, we have a projection into the central nervous system, we have a bit of processing, and we have a projection back out to muscles. If we look more closely in the brain or in the spinal cord, let's name the parts of two neurons. The direction of information flow in this process is going to be from left to right, and we have the presynaptic neuron on the right before the synapse, the postsynaptic neuron on the left after the synapse. We have the nuclei of both the presynaptic and the postsynaptic cells. We actually have 3 postsynaptic cells. We have the dendrites of both neurons. The word dendrite comes from the Greek word for tree because, in fact, the dendrites do look a little bit like trees. We have postsynaptic dendrites as well. We have the axon of one of the neurons, and in fact we have axons of the other neurons as well. The axon comes from the Greek for axis because it seems to be at the center of things, and we have a myelin sheath around the axon which makes the nerve conduct more rapidly. We have the presynaptic and postsynaptic parts of the synapse. We'll look at that in greater detail on the next slide. This is a way of getting familiar with the neurons and with the very simple 2-neuron circuit. Now, if we blow up the area where one neuron contacts another, we find the synapse, which is a point of information processing in the central nervous system. It's a little chemical hop. The nerve impulse is electrical while it's traveling within neurons. It changes to chemistry to jump across the synapse, and then it usually changes back to electricity again and flows on its way. I'll remind you that a human brain contains about 10 to the 11th neurons, and each of these might receive about 1,000 synapses apiece for a total of 10 to the 14th synapses in the brain. Most of these synapses form during the first 2 years of life. Thus, we have about 1 million synapses forming in a fetus and infant. This is a very difficult problem, which most mammals solve in developmental neurobiology. The synapse is a point of connection, of information processing, and it comes from the Greek for connection or junction. It was many years before we understood actually that there was a chemical jump in the synapse. It was not actually until the electron microscope was invented in the 40s. The first electron micrographs of synapses were shown, and they clearly showed a gap between the presynaptic and the postsynaptic neuron. So we have seen that chemistry is a language of the brain, for instance at synapses. Here we have an even larger view, a very simple view of a synapse. We have the presynaptic terminal. We have transmitter molecules in vesicles in the presynaptic terminal. That's yellow here. They are embedded in the cytosol or the intracellular environment. The synaptic cleft separates the presynaptic and the postsynaptic cells. It's about 500 angstroms wide. And then in the postsynaptic dendrite, we have receptors in the membranes of the postsynaptic dendrite. And as we learned previously, receptors are very much part of the story of drugs in the brain. And the receptors usually transduce the little chemical hop into electricity, and the nerve impulse begins again after a great deal of processing and summation by many receptors at many places on the postsynaptic cell. Next time we'll talk about electricity as the language of the brain.